All right, this one is going to be on skeletal muscle contraction. And skeletal muscle contraction takes place at the neuromuscular junction, the NMJ. Now, I will say we are focusing specifically on skeletal muscle, but this is how all muscles contract. Now, the muscle cell is controlled by the synaptic terminal of a motor neuron. So we actually have a neuron communicating with a muscle cell. And that's one of the reasons why the cell membrane of the neuron, as well as the sarcolemma of the muscle cell, are both electrically active, so that those impulses can actually transmit from one to another. Now, the impulse, obviously, the neurons do not touch the actual muscle cell, so the message has to get across that gap, just like it did with muscle or with nerve cells. And so we're using a neurotransmitter, very much like what we did with the nerve synapse. So we won't go through that again. But the neurotransmitter used for most of our skeletal muscle contractions is acetylcholine. And like I said before, the synaptic process is the same as in the neuron's synaptic transmission. Now, we do have a problem that can happen at this location, and it is a disease called botulism. And you may have actually picked up a habit, a shopping habit, that is actually related to botulism. Most people, if they go to the grocery store and they're buying a canned product, if they have a choice between a nice perfect can and one that is dented, 9 times out of 10, they're actually going to pick up the one that is perfect. And there actually is a reason for that. Back in times when the canning process, when food was just starting to be distributed more nationally, there was some problems with the canning process, and if cans got dropped, oftentimes the seal on those cans actually got compromised. And if those seals got compromised, the food inside could actually get contaminated with bacteria. And one of the bacteria that typically invaded those was the one that causes the disease of botulism. Now, what happens is these foods contain a toxin, so it's not the bacteria that actually gets people sick, it's the toxin that they make. And what it does is it prevents the release of acetylcholine, so that nerve impulse does not get transmitted to the muscle cell. This can lead to muscle paralysis, and it can be fatal. What you have to do is you do have to treat it kind of quickly, but it can be prevented. Now, the steps in a muscle contraction. First of all, the acetylcholine is released by a motor neuron, and it binds to the receptors on the cell membrane of a muscle cell, which is called the sarcolemma. Now, the sarcolemma works just like the membrane of a neuron in that it creates an action potential. And this action potential spreads along the membrane to these areas called T-tubules. They're almost like these little sinkholes, so to speak, in the cell membrane. Now, once the action potential goes down to the bottom of the T-tubules, it actually reaches an area where the T-tubule interacts with the sarcoplasmic reticulum. Now, sarcoplasmic reticulum is basically the endoplasmic reticulum of a muscle cell. And when that action potential reaches there, it causes the release of calcium. Now, this calcium then travels to the muscle fibers themselves, or to the actual filaments. And what it does is it binds to the troponin. Remember those little clips? Well, what it does is it moves that tropomyosin out of the way when the calcium is bound, and it exposes the active site on those actin filaments. With those active sites exposed, we get cross-bridging with myosin, and we actually get then the pivoting of that myosin head, which actually kind of pulls the thin filaments in towards the center of the sarcomere. So we go through this repeated process of cross-bridge binding, so actin and myosin binding, pivoting of the head, and then we detach. Now, the detachment is actually powered by ATP, our cellular form of energy. So we need energy in order for that to happen. And that produces our filament sliding. This is when our fiber actually shortens, so the Z lines come closer together. And you can actually see that here on this diagram, starting with number 1, where the acetylcholine is released up at the synaptic terminal. All right, the action potential reaches that T-tubule. Sarcoplasmic reticulum releases the calcium. Calcium goes to the actin, moves that tropomyosin out of the way. We get cross-bridge formation, and you can see contraction begins, and the arrows indicate that the sarcomere is actually shortening. Now, this video I actually have as a separate item on your DVD, so please feel free to go check that out. It actually is a very good animation, and it's very short. So what do we actually see in the sarcomere? So if we're looking at a sarcomere or a diagram of the sarcomere, what would we actually see? Well, the A band is going to stay the same width, but the Z lines move closer together, and both the H band and the I band get smaller. And you can actually see that here in the diagram. The top one is at rest, like the one we labeled, and the bottom one is with the contraction. The A band area stays the same size, but the H and the I band areas get smaller, and you can see the Z lines have moved in. Now, we obviously need to end a contraction at some point, and to do that, again, the process is going to start at the neuromuscular junction. First, what happens is acetylcholine gets broken down, and so the action potential stops. When that stops, the sarcoplasmic reticulum starts to reabsorb the calcium that it released before, so it kind of replenishes its supply. As the calcium goes away, the active sites on the actin molecules get covered back up because the troponin and tropomyosin go back to their normal spots. Because the active site gets covered up, we don't get any more cross-bridge interactions, so we don't slide inwards anymore, and the contraction ends. Now, obviously, we can't leave our sarcomere contracted like that, so they actually, the thin filaments actually slide back outwards on their own, which is the process we call relaxation that restores the resting length. And you can see those steps here outlined on this diagram. Now, with contraction, we have a couple of things I want to kind of talk about. First of all, we have rigor mortis, which I'm sure you've heard of before. Rigor mortis occurs after we're dead. And what happens is after we've died, because our cells aren't functioning, this calcium never gets reabsorbed after a certain amount of time because we don't have any more ATP left. Our body, since we're dead, isn't generating any more ATP. And once we use that up, we can't pull back the calcium. What happens is then the cross bridges can't detach, and so our muscles actually lock in a contracted position. And in fact, all of them do that, every single one. Now, this lasts about 15 to 25 hours until the enzymes that are actually part of the decomposition process come in and break down the actin and the myosin. Until they kind of start, degrading those, will they separate. And this is what coroners and other medical examiners use to help determine time of death. They can look at the amount of contraction, they can look at the amount of filament degradation by the enzymes, and that can actually help them pinpoint the actual time of death. The other one I want to talk about is a condition called muscular dystrophy. Now, this is a genetic condition, and what it does is it creates a defect in either the structural proteins or the functional proteins in muscle tissue. There are many different types of muscular dystrophy, so to talk about one specifically is kind of hard because there's many different types of muscular dystrophy. But basically, all of them have the common feature that they cause progressive muscle weakness and deterioration. Now, the one that we know the most about is a genetic one called Duchenne muscular dystrophy. And this is actually a sex-linked problem, and as such, it actually is seen most in males, and it usually starts between the ages of 3 and 7. Now, most of these boys, unfortunately, will not live very long, maybe their 20s, maybe their early 30s, but because the weakness and deterioration kind of kick in, after that point, the quality of their life goes way, way down. Now, we've talked about how muscles contract. Now, that's just simply the sarcomere. Let's talk about how we can actually make these contractions useful, and by that, we're actually going to classify our skeletal muscle contractions. The first one we're going to talk about is a twitch. And if you think about a twitch, everybody kind of can make a twitch. It is a quick, jerky response, but for our purposes, it's really not that useful. There are 3 parts to a twitch. There is the latent period where we have stimulation happening, but we don't have any actual contraction taking place yet. All of those things that we've talked about that are happening, like the action potential traveling and the calcium getting released, are happening at that point. So we actually haven't started any filament sliding yet. The contraction phase is the period of cross-bridge interaction. And then the last one is the relaxation phase, which is where the muscle returns to its resting length. Now what we can do is we can take this simple twitch though, and we can actually turn it into something useful. And to do that, we're going to actually create something called a tetanic contraction. And we have two types of tetanic contractions. We have something called incomplete tetanus and complete tetanus. Now please do not get this tetanus confused with a disease that is called tetanus. This is not harmful at all. In fact, as you're sitting there writing, you're actually using tetanic contractions. We will talk about the disease of tetanus here in a minute. In both types of tetanic contractions, we get a new stimulus arriving before the relaxation phase has ended in the twitch. So what happens is we get a second and a more powerful contraction. So it's almost kind of like adding those twitches together to turn it into something useful. Now with incomplete tetanus, we get almost peak tension and we get very rapid cycles of contraction and relaxation. And all of our normal muscular contractions are this type of tetanus. So again, as you're sitting there writing, walking, those are normal muscle contractions, and this is the kind of tetanus that we work with. Complete tetanus is where we achieve maximum tension, and it's achieved and it is sustained. There is no relaxation phase of the twitch, so we actually kind of hit the new stimulus just at the moment that the previous one is reaching its peak. It is a continuous cycle of contraction. And you can actually see that here. Letter A represents the summation of twitches that I talked about, whereas B, you can see we kind of get that up and down cycling for incomplete tetanus, and it doesn't quite reach that maximum level. Each of those arrows represents a new stimulus coming in. Whereas with complete tetanus, you can see how we have a nice smooth exponential growth there of the tension of the twitch, or of the contraction going up to maximum, sustaining maximum, and then completely dropping off. Now, let's talk about that disease of tetanus. This is actually caused by a bacterial infection. And the bacteria, it also has no relation to the normal muscle contraction. The bacteria typically live in areas where they don't get a lot of oxygen exposure. So a lot of times things that have rusted, like a rusty nail or something like that, they can actually live underneath the rust because they're not exposed to oxygen. And that's one of the reasons why small puncture wounds, especially with things that are metal, are so dangerous to get this disease of tetanus, because with a small puncture wound, you don't bleed a lot. There's not a lot of flushing out of the wound. And what it happens is that wound will actually seal over very easily, and it will trap that bacteria inside, oftentimes in an area where there's very little oxygen. So they're happy as clams. They've got a nice warm environment with a lot of nutrients, and they're protected. So they will actually start to do what the little bacteria do and create this disease. They release a toxin, and this toxin affects our motor neurons. And what they do, this toxin does, is create very sustained and powerful contractions. Where we first notice this happening is with our mouth, because we have a problem opening it. If we contract our mouth, it closes. When we relax it is when it opens. So this is where this disease gets its nickname of lockjaw. Fortunately, though, there is a vaccine available. We need booster shots every 10 years or so to keep it current. Now we have two other types of contractions to talk about that are opposites of each other. An isotonic contraction is one that produces the pivoting of a joint. So the muscle actually changes length, but as we do so, the tension stays the same. So this would be like me picking up a book. An isometric contraction, on the other hand, no pivoting takes place. So the muscle does not change length, but the tension increases and in fact maximizes. And what happens is, as this tension stays very, very high, the number of muscle fibers that get stimulated actually increases. And this is a process called recruitment. Example of an isometric contraction would be like pushing against a wall. You could do a wall sit, for instance, if you're athletic. Now, there's a couple of things with regards to muscle contractions that I want to talk about here for a second. The first one is a hernia. And a hernia is something that develops due to internal pressure in your abdominal pelvic cavity. A hernia can be an organ protruding through an abnormal opening in the muscle wall. In fact, males are susceptible to something called an inguinal hernia, and this is where the intestine gets pushed into the canal that allows different things to reach the testes. Everybody is susceptible to something called a hiatal hernia, where the portion of the stomach actually pushes up through the hole that allows the esophagus to travel through the diaphragm. This one usually requires surgery, and so does the inguinal hernia. Now, there are other places that you can also get hernias, but these are the two probably most common ones. Now, one thing that refers to contraction a lot, people talk about muscle tone. Basically, muscle tone refers to the fibers that are always active, even when the rest of them are relaxed. So there are some fibers that are always active, but they don't produce enough tension to actually move anything. So they are simply there as tensing and firming the muscle, which is what our muscle tone is. Now, people who are not active often experience atrophy, and that happens when the muscle fiber is not stimulated on a regular basis. So the fibers become smaller and weaker. They lose both size and tone, but it can be reversed if the fibers are not allowed to die. So if people have casts put on for broken bones or whatever, or if they are paralyzed, people who have casts, they take them off. Oftentimes you'll find there's differences in sizes, like if you have your arm in a cast, when you first take off your cast, you'll notice your one arm has kind of shrunk. And that's because you haven't been using those muscles that actually move those bones, because your arm has been, your wrist has been kind of held in place and not allowed to move in that direction. People who are paralyzed have to go through physical therapy in order to try to maintain their muscle tone. That's why it's so important. Between that and trying to keep their bone mass, both of those are very, very important. Okay, in the next section, we're going to talk about some different ways we can use energy with our muscle fibers, as well as talk about how exercise actually affects our muscles.